Hello, and welcome to the 2021 Allport Syndrome Foundation Allport Connect Together virtual conference. I hope you are learning more about Allport Syndrome and making connections to your fellow patients, families, caregivers, and expert Allport Syndrome healthcare professionals. My name is Andre Weinstock, and I'm an X-linked Allport Syndrome patient. I started wearing hearing aids when I was 10 years old and was on hemodialysis for 2 years when I was a college student. I've been a kidney transplant recipient for over 25 years now and work as a scientist living in Boston, Massachusetts. I volunteer on the board of directors of the Allport Syndrome Foundation and coordinate the ASF's research program. In this video, you are going to hear directly from 3 world-class scientists who you are currently funding to research diverse and impactful aspects of Allport Syndrome. And we'll hear what the outcomes of the research may mean for the future of Allport Syndrome, understanding and treatment. But first, some background. This year, in your programs, you have received a roadmap of ASF research activities. This gives a simplified version of the areas we are applying your donations today with the hopes to leverage them to larger resources for the desired outcomes in the future. Where the past is to the left, we are in the double dash line today and proceeding towards the goal in the future to the right. I want to start by looking at the diagnosis and natural history data arrows at the bottom of the map. While there are distinctions between these two topics, they are inexorably intertwined with each other, as are the outcomes of research into them. Importantly, the landscape of diagnosis and natural history is changing almost as quickly as we are researching it. And this makes this an exciting time for this work, but also presents us with new and unforeseen challenges. Not that long ago, the study of genetics was the province of genealogists and historians, not something performed in test tubes with a lab. Families were typically homogeneous, rooted in a geographical location for generations, and importantly, large. If there was a quote-unquote genetic disease in a family, it would be apparent because a certain number of siblings would exhibit the same symptoms. For a family of 8 children, with apologies to ABC, a classic Mendelian or monogenetic disease like Alport syndrome would express in 2 to 6 of the 8 kids. Probably hard not to notice. Then there are usually quite many aunts, uncles, great aunts, great uncles, and slews of cousins nearby to also make it even harder not to notice the condition. At the end of the 19th century and start of the 20th century, with the start of formalization of medical and scientific literature, some of these conditions started to be written up, and then typically named after the almost universally white male doctor who was first to publish. So, in a sense, we as a community are extremely fortunate that the South African Scottish physician A. Cecil Alport had a name that's placed at the beginning of the alphabetized list. As most of you are aware, Cecil Alport was practicing medicine in Egypt in the 1920s, then a British colony, and noted several families, large local families, with high numbers of deaths due to kidney failure, especially in the young men. He published the results of these observations in the British Medical Journal, the 19th of March, 1927, Volume 1, pages 504 to 506. And since then, the associated disease has been referred to as Allport Syndrome. Capital A, no apostrophe S after Alport, lowercase s in syndrome. Also, it's easier to say than hereditary familial congenital hemorrhagic nephritis. Fast forward to the first quarter of the 21st century, and families typically look very different than they did 100 years ago, or even 40 years ago on ABC sitcoms. Not only on the whole are they smaller, but also geographically dispersed and much more heterogeneous, with step-parents, half-siblings, second and third marriages, and adoptions. If a genetic disease exists, it may only be in one member of the extended family in a generation, and easily missed or misdiagnosed as an inherited syndrome. However, in the year 2000, the technology to sequence an entire human genome became a reality, and a new tool was born which allows us to probe the ancestry and genetic conditions of an individual independent of their family, family size, or family history. Right now, the cost to perform what is called whole exome sequencing, which examines all the genes that could potentially cause a genetic disease, is about $500, and sometimes completely covered by insurance, and can take as little as 2 weeks from performing a cheek swab to the receipt of a diagnostic risk report. So that leaves us at a point now where we are starting to have massive data banks of genetic data, server farms as the primary source of prevalence and potential diagnosis of genetic disease. Ironically, this now has to be linked back to the patients to understand what their potential risks are, and opens up new challenges of classifying who has a disease, how severe will it be for them, and what are the mitigating factors such as diet, exercise, and cultural norms. And especially, what does it mean for planning a family? These are some of the questions that our first spotlighted researcher is working on.
Hi, everyone, and thank you to the organizers for the opportunity to share a little with you about the research we're doing in Allport Syndrome. I trust you are all enjoying the meeting, and I hope that one day we can meet in person. My name is Momita Barua, and I'm an adult nephrologist at Toronto General Hospital in Toronto, Canada. First and foremost, I want to express my gratitude to the Allport Syndrome Foundation for their support of our research. My co-PI, Andrew Patterson, and I were awarded the ASF grant in 2020 for a project entitled Sex-Specific Penetrance for Predictive Diagnosis in Allport Syndrome. Our interest in Allport Syndrome was launched about 3 years ago when we conducted a study where we performed unbiased genetic testing in adult patients with a pathologic pattern of injury called FSGS. There can be many causes for this pattern of injury, but through genetic testing of over 100 genes, we discovered that approximately 5% of cases were due to undiagnosed Allport syndrome. Through my clinical experience as an adult nephrologist, I began to realize that the symptoms associated with Allport syndrome ranged from the classic symptoms that were described by Dr. Cecil Alport and what we were taught in fellowship, but also included moderate and milder phenotypes, such as later onset adult FSGS or isolated hematuria, which is blood in the urine. And these less classic forms of the disorder were vulnerable to not being diagnosed as Allport syndrome. So we wanted to understand the full clinical manifestations of the disease to improve diagnosis, but also wanted to understand the genetic factors that influences this variability from mild, moderate, and severe. Research up until recently has been biased towards studying the more severe cases with the obvious diagnosis of Allport syndrome. But there are now genetically characterized cohorts that are intended to represent the general adult population, such as one called the UK Biobank. In this cohort, approximately 500,000 adults aged 40 to 69 at the time of recruitment have had genome-wide genetic characterization that is linked to their medical records. And when we look at studies that have already been published from the UK Biobank, we see that several variants in the type 4 collagen genes causal for Allport syndrome have been linked to blood in the urine or hematuria and protein in the urine or albuminuria, which are symptoms of Allport syndrome. And so by virtue of recruitment procedures in the UK Biobank, we think that these cases are relatively mild. Based on these studies, we can start to estimate disease prevalence, and we see that at least 1 in 800 UK Biobank participants have a type 4 collagen variant that is linked to hematuria and albuminuria. And if you want to hear more, I have a 15-minute talk available on YouTube that was posted by the Allport Syndrome UK called Spotlight on Canadian Research that was posted February 17, 2021, not to be confused with an earlier podcast or talk that I did in 2020. You can also read about our research in a review paper that we published in Pediatric Nephrology in February 2021. Our ASF-funded project continues to evaluate genetic factors that influences Allport syndrome using the UK Biobank data, and we plan on sharing our results soon. Our work will help to improve diagnosis and also identify genetic factors that can be used for prognostication. So I'd like to conclude where I started. On behalf of my co-PI and study team, we are incredibly grateful to the Allport Syndrome Foundation and for all of your support. Thank you and enjoy the conference.
Returning to the research roadmap, Dr. Barua and Dr. Patterson's research falls right about here. A large number of Allport syndrome patients, maybe even the majority of Allport syndrome patients, are misdiagnosed and do not know they have Allport syndrome. However, a diagnosis of Allport syndrome is far from a one-size-fits-all. Considering X-linked, autosomal recessive, autosomal dominant, and digenic versions, the mutation variant, missense, nonsense, insertion, deletion, duplication, and so forth, kidney failure may be a certainty or may never occur, and everything in between. And then there are the other indications: severity of hearing loss and age of onset, vision problems, and even less known, problems with other organs that may be specific to just one familial variant, or universal, but in varying degrees of severity. Answering these with better understanding is critical for diagnosis and for pharmaceutical companies to know their target. They cannot formulate a drug to remedy the disease if they, or even we, do not understand all the nuances of our own condition. Now we want to go to the top of the roadmap and fundamental research. Whereas diagnosis and natural history are about looking at whole patients, whole families, and indeed all Alport patients in the world, fundamental research starts at the molecular level, within the kidney, within the inner ear, at the subcellular level. Most of you are already very familiar with the kidney, and within the kidney are the nephrons and the glomeruli in the nephrons. If we zoom down even further, we get the podocytes, endothelial cells, and the GBM, the glomerular basement membrane. The endothelial cells form a semi-porous wall of the tiniest blood vessels within the glomerulus. The podocytes kind of crawl around on the urine side of the glomerulus, and the GBM between them. The endothelial cells, the GBM, and the podocytes act together as the first line of defense filter for the kidney. Zooming down even further, we know that the collagen 4 alpha 345, the protein that is affected by having Allport syndrome, is manufactured in the podocyte, with some instructions coming from the endothelial cells and deposited as a mesh-like network to create the GBM. It is useful to think of the analogy that our next two spotlighted researchers use. The podocyte is a collagen 4 making factory. But just like any factory, it's not just a warehouse filled with the final product. There are many different machines needed to make the product, and the machines that maintain those machines, and water, air conditioning, oil, lighting, cleaning, et cetera, et cetera. In the podocyte, GBM, and endothelial cells, these machines are proteins, and there are tens of thousands of them active at any moment. Collagen 4 is formed from just 3 of these proteins. And then there are about a dozen or so other proteins that are directly responsible for making the collagen 4 or transporting it and setting it up in the basement membrane. Then there are hundreds of more proteins that support those proteins. For example, a protein that is responsible for building a part of the protein that moves the collagen 4 from inside the podocyte to outside the podocyte. Which of these tens of thousands of proteins is active at any moment in time is dependent on many factors. Are you awake or are you asleep? Are you a newborn or an adult? Are you male or female? Do you have Allport syndrome or not? And if you do have Allport syndrome, what kind? Our next spotlighted researcher is able to take a snapshot of all these thousands of proteins at a moment in time in the glomerulus of a single patient, and then mine the terabytes of data to elicit patterns of which proteins are active and in what amounts at that time. This is important because there are still thousands of proteins involved in the functioning of the glomerulus, which we still do not know what they do. And one of these just might be an answer for helping to cure Allport syndrome.
Hello, my name is Ben Humphreys, and I'm a physician scientist at Washington University in St. Louis. I'm very pleased to join you at the Allport 2021 Connect meeting to give you a brief overview of some of the research that we have been conducting with support from the Alport Foundation. The primary goal of our research is to generate a comprehensive atlas at single cell level that will describe the gene changes that occur during the full time course and progression of Allport syndrome in a mouse model. We are using a very transformative technology to create this detailed atlas. That technology is called single cell RNA sequencing, and cells are the basic fundamental unit of life. And what we are doing is tracking the changes that occur in every single cell type in the kidney, and there are over 30 different cell types during the progression of Alport disease with very high resolution. We hope that the generation of this atlas will allow us to prioritize, and indeed we have already identified specific cell types that we believe are driving progression in Alport syndrome, and analysis of these cells we are confident will reveal new therapeutic targets that someday will hopefully slow progression of Allport syndrome in patients suffering from the disease. Funding from the Alport Foundation has been absolutely critical to get this project underway because it doesn't fit under the normal kind of research topics that are funded by the National Institutes of Health, which are generally more hypothesis driven. By contrast, what we are doing with this atlas is creating a publicly available web data visualization tool, which will allow the entire Alport research community to query our very detailed data set with genes and questions and pathways that they are studying in their own labs. And in this way, we hope to contribute in a very broad way to knowledge around Alport syndrome. I'm very grateful to have received support from the Alport Foundation, and I want to thank them as well as all Alport syndrome patients, their families and communities, and I hope you enjoy the rest of this meeting.
Despite the linear format of the roadmap, in science, we never know when something that seems completely unrelated may lead to a breakthrough that is critical to our own understanding of Alport syndrome. Such is the case of a pair of researchers who were studying bacteria metabolism in the 1990s. Bacteria are a single-cell organism and don't have, per se, the systems that complex multicellular organisms have. Bacteria don't have hearts, they don't have kidneys, they don't have brains, nor do they have nerves or an immune system. However, just like us big animals and plants, bacteria also suffer from viruses called phages. So how do bacteria protect themselves from phages without an immune system? Through a lot of painstaking work over 15 years and dozens of collaborations, this pair of researchers found that bacteria, in their own DNA, keep a quick access library of all the DNA of every phage they have ever encountered. Importantly, this library and the set of proteins that allow it to function works very quickly and allows for all sorts of DNA to be cut and pasted in just the right way, like having instant access to exactly the right book at the right time. How these scientists recognize the library is that each phage DNA strand, each book, if you will, was referenced by a short cluster of palindromes in its own DNA base pairs, a short sequence of DNA that reads the same both forwards and backwards, like ATGC CGTA. They called them clustered, regularly interspersed short palindromic repeats, CRISPR for short. They also had the very key insight that this bacteriophage library immune system could work in any cell type, with any gene to very quickly and easily replace one variant with another. So a mutation in a gene that was considered deleterious to the host cell could quickly be replaced with a healthy version. This meant, suddenly, what took gene editing technology from capabilities that were only in 2011, akin to looking like a reference from a traditional physical paper library, to today, in 2021, like looking for a reference on the internet. The speed and ease of convenience is orders of magnitude, and the impact for all genetic manipulation and all species and treatment of genetic diseases is immense. This pair of researchers, Dr. Jennifer Zduna and Emmanuel Charpentier, were awarded Nobel Prizes in 2020. So now we have this new potential curative tool that wasn't on anyone's radar in human genetics only 10 years ago, suddenly appearing. While it isn't clear how CRISPR technology can be delivered into a solid organ like the kidney, it is imperative to start examining if and how CRISPR might work as a potential therapeutic for Alport syndrome. Our final spotlighted researcher is the first lab to create mice with Alport syndrome with gene editing. One model of the many he has created over the years has the ability to mimic CRISPR's potential, as if it can be delivered into the kidney. So with this research, we are taking our first steps into looking for what might be a cure for Alport syndrome.
Hello, my name is Jeff Miner. I'm a PhD scientist who has been studying Alport syndrome for almost 30 years. I am a professor in the Division of Nephrology at Washington University School of Medicine in St. Louis. I'm very pleased that the Alport Syndrome Foundation invited me to present some of my lab's ongoing research activities that is being funded by ASF. To help me do this, I'm going to share a couple of slides with you that I hope will be informative. So this first slide should look familiar. This is the kidney glomerulus, which filters the blood, or a schematic diagram of it. This is the filtration barrier here that lies between the bloodstream in these capillaries and the urinary space, which will collect what will become the final urine. There are two aspects of this I want to mention briefly. First is that one of the components of the filtration barrier is a cell called a podocyte, which for the purposes of this presentation, you should think of as a collagen 4 alpha 345 factory. This is the type 4 collagen that is defective or missing in patients with Allport syndrome. Podocytes deposit this collagen into the glomerular basement membrane, a non-cellular structure that serves as an important component of the filtration barrier and is physically defective in Allport syndrome. Over the years, my lab has generated a number of mouse genetic tools that allow us to study various unique aspects of collagen 4 and Allport syndrome biology. In this case, I'm showing you a figure from a manuscript where we report that if we start the podocyte collagen 4 factory in Allport mice, we can actually rescue their kidney disease. With no treatment, these Allport mice reach kidney failure between 15 and 35 weeks of age. But if we induce the start of the collagen 4 factory in podocytes between 0 and 3 weeks of age, then we can essentially rescue the kidney function of these mice. Only one of these animals reached kidney failure, and not until after 35 weeks of age. So it was quite effective. So having that in mind, we now know that there are tools that now and hopefully in the not too distant future will be used in patients with genetic diseases such as Allport syndrome. CRISPR is a way of doing gene repair. Viral gene delivery is a way of delivering genes to replace defective genes. There's something called nonsense mutation bypass, either by exon skipping or read-through therapy, that should be effective at bypassing certain types of mutations that cause disease. So all of these you can think of as a gene therapy type of rescue. So using the mice that we generated, the Allport Syndrome Foundation is funding studies where we are basically trying to determine what is the latest point of kidney failure or progression towards kidney failure that we can rescue the gene defect and still have an impact at slowing kidney disease progression. We hope that these results will be translatable to human patients and that someday that knowledge will allow these tools to be used at the right time in patients in order to cure their Allport syndrome. So with that, thanks for your attention, and I hope you enjoy the rest of the meeting, and I hope to have results of our studies to present to you at the next Alport Connect meeting. Thanks very much.
Thank you for your attention, and many thanks to Drs. Barua, Patterson, Humphreys, and Miner for their time and efforts. If you have questions on this presentation, for any of our spotlighted researchers, the roadmap, or about other efforts to research Allport syndrome, you can address them to me, Andre Weinstock, through the ASF community app at research@alportsyndrome.org or via the ASF website. Importantly, Lisa Bonebrake, ASF Executive Director, will be hosting a session later in this conference describing how you can actively participate in this research and become a superhero, like Annie from our Annie Has Allport Syndrome coloring books. We look forward to your attendance at that session. Please enjoy the remainder of the conference, and I'm looking forward to seeing you in person next year.